Macroevolution. Macroevolution comprises the evolutionary processes and patterns which occur at and above the species level. 1, 2, 3. In contrast, microevolution is evolution occurring within the populations of a single species. In other words, microevolution is the scale of evolution that is limited to intraspecific, within species, variation, while macroevolution extends to interspecific between species, variation. 4. The evolution of new species, speciation, is an example of macroevolution. This is the common definition for macroevolution, used by contemporary scientists. A, B, C, D, E, F, G, H, I, although the exact usage of the term has varied throughout history. 4, 10, 11. Macroevolution, evolution of species in higher taxonomic groups, genera, families, orders, etc., and uses evidence from phylogenetics. 5. The fossil record. 9. And molecular biology to answer how different taxonomic groups exhibit different species diversity and/or morphological disparity. 12. Origin and changing meaning of the term. After Charles Darwin published his book On the Origin of Species 13 in 1859, evolution was widely accepted to be a real phenomenon. However, many scientists still disagreed with Darwin that natural selection was the primary mechanism to explain evolution. Prior to the modern synthesis, during the period between the 1880s to the 1930s, dubbed the eclipse of Darwinism, many scientists argued in favor of alternative explanations. These included orthogenesis, and among its proponents was the Russian entomologist Yuri A. Filipchenko. Filipchenko appears to have been the one who coined the term macroevolution in his book Variabilität und Variation, 1927. While introducing the concept, he claimed that the field of genetics is insufficient to explain the origin of higher systematic units above the species level. In this way, modern genetics undoubtedly lifts the evolution of biotypes, Giordanones and lineones, i.e. variations within a species, a kind of microevolution, but that evolution of the higher systematic groups, which has always particularly occupied the minds of men, a kind of macroevolution, lies entirely outside its field of vision, and this circumstance seems to us only to emphasize the considerations we have given above about the lack of an inner relationship between genetics and the theory of descent, which is mainly concerned with macroevolution. State of affairs, it must be admitted that the decision of the question depends on the factors of the larger features of evolution, of what we call macroevolution, must occur independently of the results of current genetics. As advantageous as to rely on the exact results of genetics in this question, they are, in our opinion, completely useless for this purpose, since the question about the origin of the higher systematic units lies entirely outside the field research area of genetics. As a result, the latter is also an exact science, while the doctrine of descent today, as well as in the 19th century, has a speculative character. Filipchenko, Variabilität und Variation, 1927, pages 93 to 94, 11. Regarding the origin of higher systematic units, Filipchenko stated his claim that like produces like. A taxon must originate from other taxa of equivalent rank. A new species must come from an old species, a genus from an older genus, a family from another family, etc. There seems to be a fundamental misunderstanding here. At demented, it is hardly correct to see the Giordanones J as products of the fission of a lineone. J. It is even more incorrect to assume that, according to modern views, a Giordanone can or must transform into a new lineone in the process of evolution. On the contrary, it seems to us that in evolution the various taxonomic units behave in such a way that like produces like. A new biotype J arises from one biotype through mutation. A Giordanone forms a second Giordanone through a regrouping of the biotypes that make up it and the appearance of some new ones. Finally, a lineone consisting of several Giordanones splits into two independent lineones as a result of the disappearance of some of them. It is quite true that no one has observed a transformation of the races into a species, but that need not be the case, since in the process of evolution a new species or species usually arise from an old species, a new genus from another genus, etc. Yuri Filipchenko, Variabilität und Variation, 1927, page 89, 11. Filipchenko believed this was the only way to explain the origin of the major characters that define species and especially higher taxonomic groups, genera, families, orders, etc. For example, the origin of families must require the sudden appearance of new traits which are different in greater magnitude compared to the characters required for the origin of a genus or species. However, this view is no longer consistent with contemporary understanding of evolution. Furthermore, the Linnaean ranks of genus and higher are not real entities but artificial concepts which break down when they are combined with the process of evolution. 1510. Nevertheless, Filipchenko's distinction between microevolution and macroevolution had a major impact on the development of evolutionary science. The term was adopted by Filipchenko's protege Theodosius Dobzhansky in his book, Genetics und die Origin of Species, 1937, a seminal piece that contributed to the development of the modern synthesis. Macroevolution was also adopted by those who used it to criticize the modern synthesis. A notable example of this was the book The Material Basis of Evolution, 1940, by the geneticist Richard Goldschmidt, a close friend of Filipchenko. 16. Goldschmidt suggested saltational evolutionary changes either due to mutations that affect the rates of developmental processes, 17, or due to alterations in the chromosomal pattern. 18. The latter idea was widely rejected by the modern synthesis, but the hopeful monster concept based on evolutionary developmental biology, or Evo-Devo, explanations found a moderate revival in recent times. 19. Occasionally such dramatic changes can lead to novel features that survive. To saltational evolution, Dobzhansky, 21, suggested that the difference between macroevolution and microevolution reflects essentially a difference in time scales, and that macroevolutionary changes were simply the sum of microevolutionary changes over geologic time. This view is broadly accepted, and accordingly, the term macroevolution has been used widely as a neutral label for the study of evolutionary changes that take place over a very large timescale. 2. Further, species selection 2 suggests that selection among species is a major evolutionary factor that is independent from and complementary to selection among organisms. Accordingly, the level of selection has become the conceptual basis of a third definition, which defines macroevolution as evolution through selection among interspecific variation. 4. Microevolution versus macroevolution. That both micro and macroevolution, including common descent, are supported by overwhelming evidence remains uncontroversial within the scientific community. However, there has been considerable debate over the past 80 years regarding causal and explanatory connection between microevolution and macroevolution. 1. The extrapolation view holds there is no fundamental difference between the two aside from scale, i.e. macroevolution is merely cumulative microevolution. The patterns observed at the macroevolutionary scale can be explained by microevolutionary processes over long periods of time. Decoupled view holds that microevolutionary processes are decoupled from macroevolutionary processes because there are separate macroevolutionary processes that cannot be sufficiently explained by microevolutionary processes alone. Macroevolutionary processes are underlain by microevolutionary phenomena and are compatible with microevolutionary theories, but macroevolutionary studies require the formulation of autonomous hypotheses and models which must be tested using macroevolutionary evidence. In this etymologically very important sense, macroevolution is decoupled from microevolution. Macroevolution is an autonomous field of evolutionary study. Francisco J. Ayala, 1983, 23. Many scientists see macroevolution as a field of study rather than a distinct process that is similar to the process of microevolution. Thus, macroevolution is concerned with the history of life and macroevolutionary explanations encompasses ecology, paleontology, mass extinctions, plate tectonics, and unique events such as the Cambrian explosion. 24, 5, 25, 26, 16, 10, 27. The evolutionary process of changing heritable characteristics, e.g. changes in allele frequencies, is described by population genetics, with mechanisms such as mutation, natural selection, and genetic drift. However, the scope of evolution can be expanded to higher scales where different observations are made. Macroevolutionary mechanisms are provided to explain these. 2. For example, speciation can be discussed in terms of the mode, i.e. how speciation occurs. Different modes of speciation include sympatric and allopatric. Scientists research the tempo of speciation, i.e. the rate at which species change genetically and/or morphologically. Classically, competing hypotheses for the tempo of speciation include phyletic gradualism and punctuated equilibrium. Lastly, what are the causes of speciation is also extensively researched. 1. Can be asked regarding the evolution of species in higher taxonomic groups, genera, families, orders, etc., and how these have evolved across geography and vast spans of geological time. Such questions are researched from various fields of science. This makes the study of macroevolution interdisciplinary. For example, how different species are related to each other via common ancestry. This topic is researched in the field of phylogenetics. The rates of evolutionary change in across time in the fossil record. 5. Why do some groups experience a lot of change while others remain morphologically stable? The latter case are often called living fossils. However, this term is criticized for wrongly implying that such species have not evolved. The term stabilimorph has been proposed instead. 28. And causes of major events in paleontological history, including mass extinctions and evolutionary diversifications. 9. Prominent examples of mass extinctions are the Permian-Triassic and Cretaceous-Paleogene events. In contrast, famous evolutionary radiations include the Cambrian explosion and Cretaceous terrestrial revolution. Why species are high taxonomic groups even in spite of having similar ages, exhibit different survival, extinction rates, species diversity, and/or morphological disparity. The observation of long-term trends in evolution. Evolutionary trends can be passive, resembling diffusion, or driven, directional. A related question is whether these trends are directed in some way, e.g. towards complexity or simplicity. 12. Distinctive and of complex traits, which differentiate species in higher taxa from another, have evolved. Examples of this include gene duplication, heterochrony, novelty in Evo-Devo from facilitated variation, and constructive neutral evolution. Macroevolutionary processes. Speciation. According to definition, the evolutionary transition from the ancestral to the daughter species is microevolutionary, because it results from selection or, more generally, sorting among varying organisms. However, speciation has also a macroevolutionary aspect, because it produces the interspecific variation species selection operates on. Another macroevolutionary aspect of speciation is the rate at which it successfully occurs, analogous to reproductive success in microevolution. 2. Species is the process in which populations within one species change to an extent at which they become reproductively isolated, that is, they cannot interbreed anymore. However, this classical concept has been challenged and more recently, a phylogenetic or evolutionary species concept has been adopted. Their main criteria for new species is to be diagnosable and monophyletic, that is, they form a clearly defined lineage. 29-30. Darwin first discovered that speciation can be extrapolated so that species not only evolve into new species, but also into new genera, families and other groups of animals. In other words, macroevolution is reducible to microevolution through selection of traits over long periods of time. 31. In addition, some scholars have argued that selection at the species level is important as well. 32. The advent of genome sequencing enabled the discovery of gradual genetic changes both during speciation but also across higher taxa. For instance, the evolution of humans from ancestral primates or other mammals can be traced to numerous but individual mutations. 33. Evolution of new organs and tissues. One of the main questions in evolutionary biology is how new structures evolve, such as new organs. Macroevolution is often thought to require the evolution of structures that are completely new. However, fundamentally novel structures are not necessary for dramatic evolutionary change. As be seen in vertebrate evolution, most new organs are actually not new, they are simply modifications of previously existing organs. For instance, the evolution of mammal diversity in the past 100 million years has not required any major innovation. 34. All of this diversity can be explained by modification of existing organs, such as the evolution of elephant tusks from incisors. Other wings, modified limbs, feathers, modified reptile scales, 35 lungs, modified swim bladders, e.g. found in fish 36-37, or even the heart, a muscularized segment of a vein. 38. The same concept applies to the evolution of novel tissues. Even mental tissues such as bone can evolve from combining existing proteins, collagen, with calcium phosphate, specifically, hydroxyapatite. This probably happened when certain cells that make collagen also accumulated calcium phosphate to get a protobone cell. 39. Molecular macroevolution. Microevolution is facilitated by mutations, the vast majority of which have no or very small effects on gene or protein function. For instance, the activity of an enzyme may be slightly changed or the stability of a protein slightly altered. However, occasionally mutations can dramatically change the structure and functions of protein. This may be called molecular macroevolution. Metabolic enzyme galactokinase can be converted to a transcription factor in yeast by just a 2-amino acid insertion. Protein function. There are countless cases in which protein function is dramatically altered by mutations. For instance, a mutataldehyde dehydrogenase EC 1.2.1.10 can change it to a 4-hydroxy-2-oxopentanoate pyruvate lyase EC 4.1.3.39, i.e., a mutation that changes an enzyme from one to another EC class. There are only 7 main classes of enzymes. For example is the conversion of a yeast galactokinase GAL1 to a transcription factor GAL3, which can be achieved by an insertion of only 2 amino acids. 41. While some mutations may not change the molecular function of a protein significantly, their biological function may be dramatically changed. For instance, most brain receptors recognize specific neurotransmitters, but that specificity can easily be changed by mutations. This is shown by acetylcholine receptors that can be changed to serotonin or glycine receptors which actually have very different functions. Their similar gene structure also indicates that they must have arisen from gene duplications. 42. Protein structure. Although protein structures are highly conserved, sometimes one or a few mutations can dramatically change a protein. For instance, an Ig binding, 4+ fold can be transformed into an albumin binding, 3-alpha fold via a single amino acid mutation. This example also shows that such a transition can happen with neither function nor native structure being completely lost. In other words, even when multiple mutations are required to convert one protein or structure into another, the structure and function is at least partially retained in the intermediary sequences. Similarly, domains can be converted into other domains, and thus other functions. For instance, the structures of SH3 folds can evolve into OB folds which in turn can evolve into CLB folds. 44. Evolutionary faunas. Macroevolutionary benchmark study is Sepkowski's 45-46 work on marine animal diversity through the Phanerozoic. His icon of the numbers of marine families from the Cambrian to the recent illustrates the successive expansion and dwindling of 3 evolutionary faunas that were characterized by differences in origination rates and carrying capacities. Long-term ecological changes and major geological events are postulated to have played crucial roles in shaping these evolutionary faunas. 47. Stanley's rule. Macroevolution is driven by differences between species in origination and extinction rates. Remarkably, these two factors are generally positively correlated. Taxa that have typically high diversification rates also have high extinction rates. This observation has been described first by Stephen Stanley, who attributed it to a variety of ecological factors. 48. The opposite correlation of origination and extinction rates is also a prediction of the Red Queen hypothesis, which postulates that evolutionary progress, increase in fitness of any given species causes a decrease in fitness of other species, ultimately driving to extinction those species that do not adapt rapidly enough. 49. High rates of origination must therefore correlate with high rates of extinction. 4. Stanley's rule, which applies to almost all taxa and geologic ages, is therefore an indication for a dominant role of biotic interactions in macroevolution. Macromutations, single mutations leading to dramatic change. Normal phenotype. Bithorax phenotype. Mutations in the ultrabithorax gene lead to a duplication of wings in fruit flies. Examples. The vast majority of mutations are inconsequential, some can have a dramatic effect on morphology or other features of an organism. One of the best studied cases of a single mutation that leads to massive structural change is the ultrabithorax mutation in fruit flies. The mutation duplicates the wings of a fly to make it look like a dragonfly, a different order of insect. Evolution of multicellularity. The evolution of multicellular organisms is one of the major breakthroughs in evolution. The first step of converting a unicellular organism into a metazoan, a multicellular organism, is to allow cells to attach to each other. This can be achieved by one or a few mutations. In fact, many bacteria form multicellular assemblies, e.g. cyanobacteria or myxobacteria. Another species of bacteria, Junipea saculi, form well-ordered sheets of cells, which ultimately develop into a bulbous structure. Similarly, unicellular yeast cells can become multicellular by a single mutation in the ACE2 gene, which causes the cells to form a branched multicellular form. 52. Evolution of bat wings. The wings of bats have the same structural elements, bones, as any other five-fingered mammal. See periodicity in limb development. However, the finger bones in bats are dramatically elongated, so the question is how these bones became so long. It has been that certain growth factors such as bone morphogenic proteins, specifically BMP2, is overexpressed so that it stimulates an elongation of certain bones. Genetically, the bat genome identified the changes that lead to this phenotype and it has been recapitulated in mice. When specific bat DNA is inserted in the mouse genome, recapitulating these mutations, the bones of mice grow longer. 53. Limb loss in lizards and snakes. Limb loss in lizards can be observed in the genus Larista, which shows many intermediary steps with increasing loss of digits and toes. The species shown here, Larista cinerea, has no digits and only one toe left. Snakes evolved from lizards. Phylogenetic analysis shows that snakes are actually nested within the phylogenetic tree of lizards, demonstrating that they have a common ancestor. 54. This split happened about 180 million years ago and several intermediary fossils are known to document the origin. In fact, limbs have been lost in numerous clades of reptiles, and there are cases of recent limb loss. For instance, the Larista has lost limbs in multiple cases, with all possible intermediary steps. That is, there are species which have fully developed limbs, shorter limbs with 5, 4, 3, 2, 1 or no toes at all. 55. Human evolution. While evolution from their primate ancestors did not require massive morphological changes, our brain has sufficiently changed to allow human consciousness and intelligence. While the latter involves relatively minor morphological changes, it did result in dramatic changes to brain function. 56. Thus, macroevolution does not have to be morphological, it can also be functional. Evolution of viviparity in lizards. The European common lizard, Zoodoca vivipara, consists of populations that are egg-laying or live-bearing, demonstrating that this dramatic difference can even evolve within a species. Most lizards are egg-laying and thus need an environment that is warm enough to incubate their eggs. However, some species have evolved viviparity, that is, they give birth to live young, as almost all mammals do. In several of lizards, egg-laying oviparous species have evolved into live-bearing ones, apparently with very little genetic change. For instance, a European common lizard, Zoodoca vivipara, is viviparous throughout most of its range, but oviparous in the extreme southwest portion. 57-58. That is, within a single species, a radical change in reproductive behavior has happened. Similar cases from South American lizards of the genus Liolimus, which have egg-laying species at lower altitudes, but closely related viviparous species at higher altitudes, suggesting that the switch from oviparous to viviparous reproduction does not require many genetic changes. 59. Behavior, activity pattern in mice. Most animals are either active at night or during the day. However, some species switch their activity pattern from day to night or vice versa. For instance, the African striped mouse, Rhabdomys pumilio, transitioned from the ancestrally nocturnal behavior of its close relatives to a diurnal one. Genome sequencing and transcriptomics revealed that this transition was achieved by modifying genes in the rod phototransduction pathway, among others. 60. Research topics. Subjects studied within macroevolution include 61. Adaptive radiations such as the Cambrian explosion. Changes in biodiversity through time. Evo-Devo, the connection between evolution and developmental biology. Genome evolution, like horizontal gene transfer, genome fusions and endosymbiosis, and adaptive changes in genome size. Mass extinctions. Estimating diversification rates, including rates of speciation and extinction. The debate between punctuated equilibrium and gradualism. Role of development in shaping evolution, particularly such topics as heterochrony and phenotypic plasticity. See also. Microevolution. Microevolution is the change in allele frequencies that occurs over time within a population. The change is due to 4 different processes: mutation, selection, natural and artificial, gene flow and genetic drift. This change happens over a relatively short, in evolutionary terms, amount of time compared to the changes termed macroevolution. Population genetics is the branch of biology that provides the mathematical structure for the study of the process of microevolution. Ecological genetics concerns itself with observing microevolution in the wild. Typically, observable instances of evolution are examples of microevolution, for example, bacterial strains that have antibiotic resistance. Microevolution provides the raw material for macroevolution. 3. Difference from macroevolution. Macroevolution is guided by sorting of interspecific variation, species selection, as opposed to sorting of intraspecific variation in microevolution. 3. Species selection occurs, affect macroevolution, where organism-level traits, aggregate traits, affect speciation and extinction rates, and b. Strict sense species selection, where species-level traits, e.g. geographical range, affect speciation and extinction rates. Evolution does not produce evolutionary novelties, but it determines their proliferation within the clades in which they evolved, and it adds species-level traits as non-organismic factors of sorting to this process. 3. 4 processes. Mutation. Duplication of part of a chromosome. Changes in the DNA sequence of a cell's genome and are caused by radiation, viruses, transposons and mutagenic chemicals, as well as errors that occur during meiosis or DNA replication. 6. 7. Errors are introduced particularly often in the process of DNA replication, in the polymerization of the second strand. These errors can also be induced by the organism itself, by cellular processes such as hypermutation. Mutations can affect the phenotype of an organism, especially if they occur within the protein coding sequence of a gene. Error rates are usually very low, 1 error in every 10 to 100 million bases, due to the proofreading ability of DNA polymerases. Without proofreading, error rates are 1,000-fold higher. Because many viruses rely on DNA and RNA polymerases that lack proofreading ability, they experience higher mutation rates. Processes that increase changes in DNA are called mutagenic. Mutagenic chemicals promote errors in DNA replication, often by interfering with the structure of base pairing, while UV radiation induces mutations by causing damage to the DNA structure. Damage to DNA occurs naturally as well, and cells use DNA repair mechanisms to repair mismatches and breaks in DNA. Nevertheless, the repair sometimes fails to return the DNA to its original sequence. Organisms that use chromosomal crossover to exchange DNA and recombine genes, errors in alignment during meiosis can also cause mutations. Errors in crossover are especially likely when similar sequences cause partner chromosomes to adopt a mistaken alignment, making some regions in genomes more prone to mutating in this way. These errors create large structural changes in DNA sequence, duplications, inversions or deletions of entire regions, or the accidental exchanging of whole parts between different chromosomes called translocation. Mutation result in several different types of change in DNA sequences. These can either have no effect, alter the product of a gene, or prevent the gene from functioning. Studies in the fly Drosophila melanogaster suggest that if a mutation changes a protein produced by a gene, this will probably be harmful, with about 70% of these mutations having damaging effects, and the remainder being either neutral or weakly beneficial. Due to the damaging effects that mutations can have on cells, organisms have evolved mechanisms such as DNA repair to remove mutations. 5. Therefore, optimal mutation rate for a species is a trade-off between costs of a high mutation rate, such as deleterious mutations, and the metabolic costs of maintaining systems to reduce the mutation rate, such as DNA repair enzymes. Viruses that use RNA as their genetic material have rapid mutation rates, 14, which can be an advantage since these viruses will evolve constantly and rapidly, and thus evade the defensive responses of e.g. the human immune system. 15. Mutations can involve large sections of DNA becoming duplicated, usually through genetic recombination. 16. These duplications are a major source of raw material for evolving new genes, with tens to hundreds of genes duplicated in animal genomes every million years. 17. Most genes belong to larger families of genes of shared ancestry. 18. Genes are produced by several methods, commonly through the duplication and mutation of an ancestral gene, or by recombining parts of different genes to form new combinations with new functions. 20. Here, domains act as modules, each with a particular and independent function, that can be mixed together to produce genes encoding new proteins with novel properties. For example, the human eye uses 4 genes to make structures that sense light, 3 for color vision and 1 for night vision, all 4 arose from a single ancestral gene. 22. Another advantage of duplicating a gene, or even an entire genome, is that this increases redundancy. This allows one gene in the pair to acquire a new function while the other copy performs the original function. 23-24 Other types of mutation occasionally create new genes from previously non-coding DNA. 25-26 Selection. Selection is the process by which heritable traits that make it more likely for an organism to survive and successfully reproduce become more common in a population over successive generations. It is sometimes valuable to distinguish between naturally occurring selection, natural selection, and selection that is a manifestation of choices made by humans, artificial selection. This distinction is rather diffuse. Natural selection is nevertheless the dominant part of selection. Natural selection of a population for dark coloration. The natural genetic variation within a population of organisms means that some individuals will survive more successfully than others in their current environment. Factors which affect reproductive success are also important, an issue which Charles Darwin developed in his ideas on sexual selection. Natural selection acts on the phenotype, or the observable characteristics of an organism, but the genetic heritable basis of any phenotype which gives a reproductive advantage will become more common in a population, see allele frequency. Over time, this process can result in adaptations that specialize organisms for particular ecological niches and may eventually result in the speciation, the emergence of new species. Natural selection is one of the cornerstones of modern biology. The term was introduced by Darwin in his groundbreaking 1859 book On the Origin of Species, 27, in which natural selection was described by analogy to artificial selection, a process by which animals and plants with traits considered desirable by human breeders are systematically favored for reproduction. The concept of natural selection was originally developed in the absence of a valid theory of heredity. At the time of Darwin's writing, nothing was known of modern genetics. The union of traditional Darwinian evolution with subsequent discoveries in classical and molecular genetics is termed the modern evolutionary synthesis. Natural selection remains the primary explanation for adaptive evolution. Genetic drift. Simulation of random genetic drift of a single given allele with an initial frequency distribution 0.5 measured over the course of 50 generations, repeated in 3 reproductively synchronous populations of different sizes. In general, alleles drift to loss or fixation frequency of 0.0 or 1.0 significantly faster in smaller populations. Genetic drift is the change in the relative frequency in which a gene variant allele occurs in a population due to random sampling. That is, the alleles in the offspring in the population are a random sample of those in the parents. And chance has a role in determining whether a given individual survives and reproduces. A population's allele frequency is the fraction or percentage of its gene copies compared to the total number of gene alleles that share a particular form. 28 Genetic drift is an evolutionary process which leads to changes in allele frequencies over time. It may cause gene variants to disappear completely, and thereby reduce genetic variability. Natural selection, which makes gene variants more common or less common depending on their reproductive success. 29 The changes due to genetic drift are not driven by environmental or adaptive pressures, and may be beneficial, neutral, or detrimental to reproductive success. The effect of genetic drift is larger in small populations, and smaller in large populations. Vigorous debates wage among scientists over the relative importance of genetic drift compared with natural selection. Fisher held the view that genetic drift plays at the most a minor role in evolution, and this remained the dominant view for several decades. In 1968 Moto Kimura rekindled the debate with his neutral theory of molecular evolution which claims that most of the changes in the genetic material are caused by genetic drift. The prediction neutral theory, based on genetic drift, do not fit recent data on whole genomes well. These data suggest that the frequencies of neutral alleles change primarily due to selection at linked sites, rather than due to genetic drift by means of sampling error. 31. Gene flow. Gene flow is the exchange of genes between populations, which are usually of the same species. 32 Examples of gene flow within a species include the migration and then breeding of organisms, or the exchange of pollen. Gene transfer between species includes the formation of hybrid organisms and horizontal gene transfer. Migration into or out of a population can change allele frequencies, as well as introducing genetic variation into a population. Immigration may add new genetic material to the established gene pool of a population. Conversely, emigration may remove genetic material. As reproduction between two diverging populations are required for the populations to become new species, gene flow may slow this process by spreading genetic differences between the populations. Gene flow hindered by mountain ranges, oceans and deserts or even man-made structures such as the Great Wall of China, which has hindered the flow of plant genes. 33. Depending on how far two species have diverged since their most recent common ancestor, it may still be possible for them to produce offspring, as with horses and donkeys mating to produce mules. 34. Such hybrids are generally infertile, due to the two different sets of chromosomes being unable to pair up during meiosis. In this case, closely related species may regularly interbreed, but hybrids will be selected against and the species will remain distinct. However, viable hybrids are occasionally formed and these new species can either have properties intermediate between their parent species, or possess a totally new phenotype. 35. Importance of hybridization in developing new species of animals is unclear, although cases have been seen in many types of animals, 36 with the gray tree frog being a particularly well-studied example. 37. Hybridization, however, an important means of speciation in plants, since polyploidy, having more than 2 copies of each chromosome, is tolerated in plants more readily than in animals. 38-39. Polyploidy is important in hybrids as it allows reproduction, with the two different sets of chromosomes each being able to pair with an identical partner during meiosis. Polyploid hybrids also have more genetic diversity, which allows them to avoid inbreeding depression in small populations. 41. Horizontal gene transfer is the transfer of genetic material from one organism to another organism that is not its offspring. This is most common among bacteria. In medicine, this contributes to the spread of antibiotic resistance, as when one bacteria acquires resistance genes it can rapidly transfer them to other species. 43. Horizontal transfer of genes from bacteria to eukaryotes such as the yeast Saccharomyces cerevisiae and the Adzuki bean beetle Calosobrichus chinensis may also have occurred. 44-45. An example of larger-scale transfers are the eukaryotic bdelloid rotifers, which appear to have received a range of genes from bacteria, fungi, and plants. 46. Viruses can also carry DNA between organisms, allowing transfer of genes even across biological domains. 47. Large-scale gene transfer has also occurred between the ancestors of eukaryotic cells and prokaryotes, during the acquisition of chloroplasts and mitochondria. 48. Gene flow is the transfer of alleles from one population to another. Migration into or out of a population may be responsible for a marked change in allele frequencies. Immigration may also result in the addition of new genetic variants to the established gene pool of a particular species or population. There are a number of factors that affect the rate of gene flow between different populations. One of the significant factors is mobility, as greater mobility of an individual tends to give it greater migratory potential. Animals tend to be more mobile than plants, although pollen and seeds may be carried great distances by animals or wind. Gene flow between two populations can also lead to a combination of the two gene pools, reducing the genetic variation between the two groups. It is for this reason flow strongly acts against speciation, by recombining the gene pools of the groups, and thus, repairing the developing differences in genetic variation that would have led to full speciation and creation of daughter species. For example, if a species of grass grows on both sides of a highway, pollen is likely to be transported from one side to the other and vice versa. If able to fertilize the plant where it ends up and produce viable offspring, then the alleles in the pollen have effectively been able to move from the population on one side of the highway to the other. Origin and extended use of the term. Origin. The term evolution was first used by botanist Robert Greenleaf Levitt in the journal Botanical Gazette in 1909, addressing what he called the mystery of how formlessness gives rise to form. 49. The production of formlessness in the egg-derived individual, the multiplication of parts and the orderly creation of diversity among them, in an actual evolution, of which anyone may ascertain the facts, but of which no one has dissipated the mystery in any significant measure. This evolution forms an integral part of the grand evolution problem and lies at the base of it, so that we shall have to understand the minor process before we can thoroughly comprehend the more general one. However, Levitt was using the term to describe what we would now call developmental biology. It was not until Russian entomologist Yuri Filipchenko used the terms macroevolution and microevolution in 1927 in his German-language work, Variabilität und Variation, that it attained its modern usage. The term was brought into the English-speaking world by Filipchenko's student Theodosius Dobzhansky in his book Genetics and the Origin of Species, 1937. 1. Use in creationism. In young Earth creationism and baraminology, a central tenet is that evolution can explain diversity in a limited number of created kinds which can interbreed, which they call microevolution, while the formation of new kinds, which they call macroevolution, is impossible. 50-51. This acceptance of microevolution, only within a kind, is also typical of old Earth creationism. 52. Scientific organizations such as the American Association for the Advancement of Science describe microevolution as small-scale change within species, and macroevolution as the formation of new species, but otherwise not being different from microevolution. In macroevolution, an accumulation of microevolutionary changes leads to speciation. 50. The main difference between the two processes is that one occurs within a few generations, whilst the other takes place over thousands of years, i.e. a quantitative difference. 4. Essentially they describe the same process. Although evolution beyond the species level results in beginning and ending generations which could not interbreed, the intermediate generations could. Creationism argue that changes in the number of chromosomes can be accounted for by intermediate stages in which a single chromosome divides in generational stages, or multiple chromosomes fuse, and cite the chromosome difference between humans and the other great apes as an example. 5. Creationists insist that since the actual divergence between the other great apes and humans was not observed, the evidence is circumstantial. Describe fundamental similarity between macro and microevolution in his authoritative textbook, Evolutionary Biology, biologist Douglas Futuyma writes. One of the most parts of the theory forged during the evolutionary synthesis of the 1930s and 1940s was that macroevolutionary differences among organisms, those that distinguish higher taxa, arise from the accumulation of the same kinds of genetic differences that are found within species. Opponents believe that macroevolution is qualitatively different from microevolution within species and is based on a totally different kind of genetic and developmental patterning. Genetic studies of species differences have decisively disproved this claim. Differences between species in quality, figure, and the processes that underlie reproductive isolation all have the same genetic properties as variation within species. They occupy consistent chromosomal positions, they may be polygenic or based on few genes, they may display additive, dominant, or epistatic effects, and they can in some instances be traced to specifiable differences in proteins or DNA nucleotide sequences. The degree of reproductive isolation between populations, whether prezygotic or postzygotic, varies from little or none to complete. Thus, reproductive isolation, like the divergence of any other character, evolves in most cases by the gradual substitution of alleles in populations. Douglas Futuyma, Evolutionary Biology, 1998 pp. 4778-56. Contrary to the claims of some anti-evolution proponents, evolution of life forms beyond the species level, i.e. speciation, has indeed been observed and documented by scientists on numerous occasions. 57. Science, creationists accepted speciation as occurring within a created kind, or Barrowman, but objected to what they called third-level macroevolution of a new genus or higher rank in taxonomy. Ambiguity in the ideas as to where to draw a line on species, created kinds, and what events and lineages fall within the rubric of microevolution or macroevolution. 58. See also. Fluctuated equilibrium due to gene flow, major evolutionary changes may be rare. Biological species concept. Group of animals capable of interbreeding. The biological species concept gives an explanation of how species form speciation. A biological species is a group of individuals that can breed together panmixia. However, they cannot breed with other groups. In other words, the group is reproductively isolated from other groups. 1-2. The words reproductively isolated are the key words of the biological species definition. Ernst Mayr, 2p273. According to Ernst Mayr, a new species forms when an existing species splits. A similar idea had been suggested in the 19th century by Moritz Wagner. Dobzhansky described the role of reproductive isolation in the formation of new species. 5. Once a species lives in two different areas, the geographical isolation makes breeding between the groups reduce or stop. Each group develops features which make breeding between them work less well. Eventually, each group becomes a good biological species, because the two species do not reproduce with each other even when they are together. This is still considered the most common reason for species splitting, and has the technical name of allopatric speciation. It is contrasted with sympatric speciation where speciation takes place even though all the members live in the same area. 6. Its role in taxonomy. The biological species is the main reason for classifying living things, but applying it in practice is not easy. Good examples of this are the following. Lack of mating, sterility or inviability in interstrain crosses has never been taken on its own as good evidence of separate species. 7. Including even Dobzhansky's own studies of sibling Drosophila species. 8. The biological species concept 9 has no bearing on asexual organisms, and has severe limitations in its application to other organisms. 8. 10. Time and effort can be expended on discussing the species concept, and devising yet further modifications and variations on proposed specific definitions. Such debates have great merit in encouraging more objective reasoning about evolution and taxonomy but, in the meantime, human predation and destruction of habitat continue to erase the subject species. Time is against us. Different species concepts produce groups of subspecies, so it is futile to contemplate a definitive taxonomic system. 11. What this is saying is that, in practice, the biological species concept is not sufficient by itself to decide on the classification of animals. 12. Historical developments. John Ray. In 1686 John Ray introduced a non-evolutionary biological concept. To him, species were distinguished by always producing the same species, and this was fixed and permanent, though considerable variation was possible within a species. 13-14. The idea of a species as a physical type of organism had a long history. This survives as the concept of a type specimen in taxonomy. It's the way Linnaeus worked in his binomial classification, and is still useful for amateur naturalists today. Charles Darwin. In The Origin, Charles Darwin said that species were labels which experts gave on the basis of their observations. Quote, I look at the term species as one arbitrarily given for the sake of convenience to a set of individuals closely resembling each other. 15. But 20 years before, he had a much better idea. He thought of species as maintained by reproductive isolation. He even says, hence species may be good ones and differ scarcely at all in any external character. Here he quotes the two sibling species of leaf warbler discovered in England by Gilbert White in 1768. At this early stage in his career, Darwin came very close to the modern biological species concept. 2p266. Modern era. In the last 70 years, two ideas have dominated the way professional biologists think about species. The first is the population genetics concept. This is where a species is seen as a group which can mate together even though they are all to some extent different. It amounts to saying that a species is a gene pool. 16. Second is the use of DNA sequence analysis to show whether similar-looking species are genetically different from each other. This is especially useful when it is not practical to do breeding experiments. Sibling species. Sibling species are often called cryptic, hidden species because their differences can only be seen by analyzing their DNA. They are very common in the marine environment. 17-18. Many cryptic species exist in all habitats. In the Rhizocelbrella hyalina, 19th DNA sequence analysis was used to show that more than 10 ecologically distinct species that had been diverging for many millions of years. Evidence from the identification of cryptic species means that older estimates of global species richness are too low. For example, mitochondrial DNA research suggests there are at least 11 genetically distinct populations of giraffes. 21. Similarly, the Amazonian frog Eleutherodactylus akindani is at least 3 different species which diverged over 5 million years ago. 22. References. 1. Meyer, Ernst 2001. Adaptive radiation. In evolutionary biology, adaptive radiation is a process in which organisms diversify rapidly from an ancestral species into a multitude of new forms, particularly when a change in the environment makes new resources available, alters biotic interactions or opens new environmental niches. 1. 2. Starting with a single ancestor, this process results in the speciation and phenotypic adaptation of an array of species exhibiting different morphological and physiological traits. The prototypical example of adaptive radiation is finch speciation on the Galapagos, Darwin's finches, but examples are known from around the world. 4 of 14 finch species found in the Galapagos archipelago, which are thought to have evolved via an adaptive radiation that diversified their beak shapes, enabling them to exploit different food sources. Characteristics. 4 features can be used to identify an adaptive radiation. 2. 1. A common ancestry of component species, specifically a recent ancestry. Note that this is not the same as a monophily in which all descendants of a common ancestor are included. 2. A phenotype-environment correlation, a significant association between environments and the morphological and physiological traits used to exploit those environments. 3. Trait utility, the performance or fitness advantages of trait values in their corresponding environments. 4. Rapid speciation, presence of one or more bursts in the emergence of new species around the time that ecological and phenotypic divergence is underway. Adaptive radiations are thought to be triggered by an ecological opportunity, 3, or a new adaptive zone. Sources of ecological opportunity can be the loss of antagonists, competitors or predators, the evolution of a key innovation, or dispersal to a new environment. Any one of these ecological opportunities has the potential to result in an increase in population size and relaxed stabilizing, constraining selection. As genetic diversity is positively correlated with population size, 5, the expanded population will have more genetic diversity compared to the ancestral population. With reduced stabilizing selection, phenotypic diversity can also increase. In addition, intraspecific competition will increase, promoting divergent selection to use a wider range of resources. This ecological release provides the potential for ecological speciation and thus adaptive radiation. 3. Occupying a new environment might take place under the following conditions. 6. Adaptive radiations are thought to be triggered by an ecological opportunity, 3, or a new adaptive zone. Sources of ecological opportunity can be the loss of antagonists, competitors or predators, the evolution of a key innovation, or dispersal to a new environment. Any one of these ecological opportunities has the potential to result in an increase in population size and relaxed stabilizing, constraining selection. As genetic diversity is positively correlated with population size, 5, the expanded population will have more genetic diversity compared to the ancestral population. With reduced stabilizing selection, phenotypic diversity can also increase. In addition, intraspecific competition will increase, promoting divergent selection to use a wider range of resources. This ecological release provides the potential for ecological speciation and thus adaptive radiation. 3. Occupying a new environment might take place under the following conditions. 6. 1. A new habitat has opened up. A volcano, for example, can create new ground in the middle of the ocean. This is the case in places like Hawaii and the Galapagos. For aquatic species, the formation of a large new lake habitat could serve the same purpose. The tectonic movement that formed the East African Rift, ultimately leading to the creation of the Rift Valley Lakes, is an example of this. An extinction event could effectively achieve this same result, opening up niches that were previously occupied by species that no longer exist. 2. This new habitat is relatively isolated. When a volcano erupts on the mainland and destroys an adjacent forest, it is likely that the terrestrial plant and animal species that used to live in the destroyed region will recolonize without evolving greatly. However, if a newly formed habitat is isolated, the species that colonize it will likely be somewhat random and uncommon arrivals. 3. The new habitat has a wide availability of niche space. The rare colonist can only adaptively radiate into as many forms as there are niches. 4. Relationship between mass extinctions and mass adaptive radiations. Phanerozoic biodiversity is shown by the fossil record. Conditions. All genera. Well-defined, genera trend line. 5. Mass extinctions. Other mass extinctions million years ago, thousands of. A 2020 found there to be no direct causal relationship between the proportionally most comparable mass radiations and extinctions in terms of co-occurrence of species, substantially challenging the hypothesis of creative mass extinctions. 7, 8, 9. Examples. Darwin's finches. Darwin's finches on the Galapagos Islands are a model system for the study of adaptive radiation. 10. Today represent approximately 15 species. Darwin's finches are Galapagos endemics famously adapted for a specialized feeding behavior, although one species, the Cocos finch Paneroloxia zanornata, is not found in the Galapagos but on the island of Cocos south of Costa Rica. Darwin's finches are not actually finches in the true sense, but are members of the tanager family Thropidae, and are derived from a single ancestor that arrived in the Galapagos from mainland South America perhaps just 3 million years ago. 12. Excluding the Cocos finch, each species of Darwin's finch is generally widely distributed in the Galapagos and fills the same niche on each island. For the ground finches, this niche is a diet of seeds, and they have thick bills to facilitate the consumption of these hard materials. 11. The ground finch is of a particular size. The large ground finch Geospiza magnirostrus is the largest species of Darwin's finch and has the thickest beak for breaking open the toughest seeds. The small ground finch Geospiza fuliginosa has a smaller beak for eating smaller seeds, and the medium ground finch Geospiza fortis has a beak of intermediate size for optimal consumption of intermediately sized seeds relative to G. magnirostrus and G. fuliginosa. 11. Some overlap. For example, the most robust medium ground finches could have beaks larger than those of the smallest large ground finches. 11. This overlap, it can be difficult to tell the species apart by eye, though their songs differ. 11. These three species often occur sympatrically, and during the rainy season in the Galapagos when food is plentiful, they specialize little and eat the same, easily accessible foods. 11. It was understood why their beaks were so adapted until Peter and Rosemary Grant studied their feeding behavior in the long dry season, and discovered that when food is scarce, the ground finches use their specialized beaks to eat the seeds that they are best suited to eat and thus avoid starvation. 11. The other finches in the Galapagos are similarly uniquely adapted for their particular niche. The cactus finches, Geospiza sp. Some longer beaks than the ground finches that serve the dual purpose of allowing them to feed on Opuntia cactus nectar and pollen while these plants are flowering, but on seeds during the rest of the year. 11. The warbler finches, Certhidia sp. have short, pointed beaks for eating insects. 11. The wood finch, Camarynchus pallidus, has a slender beak which it uses to pick at wood in search of insects. It also uses small sticks to reach insect prey inside the wood, making it one of the few animals that use tools. 11. The mechanism by which the finches initially diversified is still an area of active research. 1. The finches were able to have a non-adaptive, allopatric speciation event on separate islands in the archipelago, such that when they reconverged on some islands, they were able to maintain reproductive isolation. 2. Once they occurred in symmetry, niche specialization was favored so that the different species competed less directly for resources. 12. This second, sympatric event was adaptive radiation. 12. Cichlids of the African Great Lakes. The haploid cichlid fishes in the Great Lakes of the East African Rift, particularly in Lake Tanganyika, Lake Malawi, and Lake Victoria, form the most speciose-modern example of adaptive radiation. 13, 14, 15. These lakes are believed to be home to about 2,000 different species of cichlid, spanning a wide range of ecological roles and morphological characteristics. Cichlids in these lakes fill nearly all of the roles typically filled by many fish families, including those of predators, scavengers, and herbivores, with varying dentitions and head shapes to match their dietary habits. 15. In each case, the radiation events are only a few million years old, making the high level of speciation particularly remarkable. 15. 14. Several factors could be responsible for this diversity. The availability of a multitude of niches probably favored specialization, as few other fish taxa are present in the lakes, meaning that sympatric speciation was the most probable mechanism for initial specialization. 13. Also, continual changes in the water level of the lakes during the Pleistocene, which often turned the largest lakes into several smaller ones, could have created the conditions for secondary allopatric speciation. 15. 13. Tanganyika cichlids. Tanganyika is the site from which nearly all the cichlid lineages of East Africa, including both riverine and lake species, originated. 17. Thus, the species in the lake constitute a single adaptive radiation event but do not form a single monophyletic clade. 17. Lake Tanganyika also the least speciose of the three largest African Great Lakes, with only around 200 species of cichlid. 14. However, these cichlids are more morphologically divergent and ecologically distinct than their counterparts in Lakes Malawi and Victoria, an artifact of Lake Tanganyika's older cichlid fauna. Lake Tanganyika itself is believed to have formed 9 to 12 million years ago, putting a recent cap on the age of the lake's cichlid fauna. 14. Many of Tanganyika's cichlids live very specialized lifestyles. Gender emperor cichlid Bolingarochromis microlepis is a piscivore, often ranked the largest of all cichlids, though it competes for this title with South America's Cichlid tamensis, the speckled peacock base. 14. It is thought that giant cichlids spawn only a single time, breeding in their third year and defending their young until they reach a large size, before dying of starvation sometime thereafter. 3 species of Altalamprologus are also piscivores, but with laterally compressed bodies and thick scales enabling them to chase prey into thin cracks in rocks without damaging their skin. 14. Plecostrelini has evolved large, strangely curved teeth that are designed to scrape scales off of the sides of other fish, scales being its main source of food. 13. Gnathocromis permaxillaris possesses a large mouth with a protruding upper lip, and feeds by opening this mouth downward onto the sandy lake bottom, sucking in small invertebrates. A number of Tanganyika's cichlids are shell brooders, meaning that mating pairs lay and fertilize their eggs inside of empty shells on the lake bottom. 14. Lamprocaliteris is a unique egg-brooding species, with 15 cm long males amassing collections of shells and guarding them in the hopes of attracting females, about 6 cm in length, to lay eggs in these shells. 14. These males varies from 3 types of rival. 1. Other dominant males looking to steal shells. 2. Younger, sneaker, males looking to fertilize eggs in a dominant male's territory. And 3. Tiny, 2 to 4 cm, parasitic dwarf, males that also attempt to rush in and fertilize eggs in the dominant male's territory. These parasitic dwarf males never grow to the size of dominant males, and the male offspring of dominant and parasitic dwarf males grow with 100% fidelity into the form of their fathers. 14. A number of other highly specialized Tanganyika cichlids exist aside from these examples, including those adapted for life in open lake water up to 200 meters deep. 14. Malawi cichlids. The cichlids of Lake Malawi constitute a species flock of up to 1,000 endemic species. 15. Only cichlid species in Lake Malawi are not a part of the species flock. The eastern happy Astatilapia calitera, the Sungwa, Serenachromis robustus, and 5 tilapia species genera Oreochromis and Coptodon. All of the other cichlid species in the lake are descendants of a single original colonist species, which itself was descended from Tanganyikan ancestors. 17. The common ancestor of Malawi's species flock is believed to have reached the lake 3.4 million years ago at the earliest, making Malawi cichlids' diversification into their present numbers particularly rapid. 15. Malawi's cichlids span a similarly range of feeding behaviors to those of Tanganyika, but also show signs of a much more recent origin. For example, the Malawi species flock are mouth brooders, meaning the female keeps her eggs in her mouth until they hatch. In almost all species, the eggs are also fertilized in the female's mouth, and in a few species, the females continue to guard their fry in their mouth after they hatch. 15. Males of most species display predominantly blue coloration when mating. However, particularly divergent species are known from Malawi, including the Piscivorous Nimbochromis livingtoni, which lies on its side in the substrate until small cichlids, perhaps drawn to its broken white patterning, come to inspect the predator, at which point they are swiftly eaten. 15. Victoria's cichlids. Lake Victoria's cichlids are also a species flock, once composed of some 500 or more species. The deliberate introduction of the Nile perch, Lates niloticus, in the 1950s proved disastrous for Victoria cichlids, and the collective biomass of the Victoria cichlid species flock has decreased substantially and an unknown number of species have become extinct. However, the original range of morphological and behavioral diversity seen in the lake's cichlid fauna is still mostly present today, if endangered. These again include cichlids specialized for niches across the trophic spectrum, as in Tanganyika and Malawi, but again, there are standouts. Victoria is home to many piscivorous cichlid species, some of which feed by sucking the contents out of mouth-brooding females' mouths. Victoria's cichlids constitute a far younger radiation than even that of Lake Malawi, with estimates of the age of the flock ranging from 200,000 years to as little as 14,000. 13. Adaptive radiation in Hawaii. Aniiwi Drapanus coccinea. Note the long, curved beak for sipping nectar from tubular flowers. Has served as the site of a number of adaptive radiation events, owing to its isolation, recent origin, and large land area. The most famous examples of these radiations are presented below, though insects like the Hawaiian drosophilid flies and Hyposmocoma moths have also undergone adaptive radiation. 1920. Hawaiian honeycreepers. The Hawaiian honeycreepers form a large, highly morphologically diverse species group of birds that began radiating in the early days of the Hawaiian archipelago. While today 18 species are known to persist in Hawaii, 3 more may or may not be extinct. There were more than 50 species prior to Polynesian colonization of the archipelago. Between 18 and 21 species have gone extinct since the discovery of the islands by Westerners. The Hawaiian honeycreeper have beaks, which are specialized to satisfy a wide range of dietary needs. For example, the beak of the Achiopalaau, Hemignathus wilsoni, is characterized by a short, sharp lower mandible for scraping bark off of trees, and the much longer, curved upper mandible is used to probe the wood underneath for insects. Meanwhile, the Aniiwi Drapanus coccinea has a very long curved beak for reaching nectar deep in lobelia flowers. An entire clade of Hawaiian honeycreepers, the tribe Sidirostrini, is composed of thick-billed, mostly seed-eating birds, like the Laysan finch Telespisa cantans. 19. In some cases, similar morphologies and behaviors appear to have evolved convergently among the Hawaiian honeycreepers. For example, the short, pointed beaks of Loxops and Oreomystus evolved separately despite once forming the justification for lumping the two genera together. The Hawaiian honeycreepers are believed to have descended from a single common ancestor some 15 to 20 million years ago, though estimates range as low as 3.5 million years. 22. Hawaiian silver swords. A mixture of blooming and non-blooming Haleakala silver swords, Argyroxiphium sandwichense macrocephalum. Adaptive radiation is not a strictly vertebrate phenomenon, and examples are also known from among plants. The most example of adaptive radiation in plants is quite possibly the Hawaiian silver swords, named for alpine desert-dwelling Argyroxiphium species with long, silvery leaves that live for up to 20 years before growing a single flowering stalk and then dying. The Silver Sword Alliance consists of 28 species of Hawaiian plants which, aside from the namesake silver swords, includes trees, shrubs, vines, cushion plants, and more. The Silver Sword Alliance is believed to have originated in Hawaii no more than 6 million years ago, making this one of Hawaii's youngest adaptive radiation events. This means that the Silver Swords evolved on Hawaii's modern high islands, and descended from a single common ancestor that arrived on Kauai from western North America. 22. The closest modern relatives of the Silver Swords today are California tarweeds of the family Astraceae. 22. Hawaiian lobelioids. Hawaii is also the site of a separate major floral adaptive radiation event, the Hawaiian lobelioids. The Hawaiian lobelioids are significantly more speciose than the silver swords, perhaps because they have been present in Hawaii for so much longer. They descended from a single common ancestor who arrived in the archipelago up to 15 million years ago. Today the Hawaiian lobelioids form a clade of over 125 species, including succulents, trees, shrubs, epiphytes, etc. 23. Many species have been lost to extinction and many of the surviving species endangered. Caribbean anoles. Anole lizards are distributed broadly in the New World, from the southeastern U.S. to South America. With over 400 species currently recognized, often placed in a single genus, Anolis, they constitute one of the largest radiation events among all lizards. And radiation on the mainland has largely been a process of speciation, and is not adaptive to Hawaiian lobelioids. Hawaii is also the site of a separate major floral adaptive radiation event, the Hawaiian lobelioids. Hawaiian lobelioids are significantly more speciose than the silver swords, perhaps because they have been present in Hawaii for so much longer. They descended from a single common ancestor who arrived in the archipelago up to 15 million years ago. 22. Today the Hawaiian lobelioids form a clade of over 125 species, including succulents, trees, shrubs, epiphytes, etc. 23. Many species have been lost to extinction and many of the surviving species endangered. Caribbean anoles. Anole lizards are distributed broadly in the New World, from the southeastern U.S. to South America. With over 100 species currently recognized, often placed in a single genus, Anolis, they constitute one of the largest radiation events among all lizards. 24. Radiation on the mainland has largely been a process of speciation, and is not adaptive to any great degree, but anoles on each of the Greater Antilles, Cuba, Hispaniola, Puerto Rico, and Jamaica, have adaptively radiated in separate, convergent ways. 25. On each of these islands, anoles have evolved with such a consistent set of morphological adaptations that each species can be assigned to one of 6 ecomorphs: trunk ground, trunk crown, grass bush, crown giant, twig, and trunk. 25. Example crown giants from each of these islands: the Cuban Anolis luteogularis, Hispaniola's Anolis ricordi, Puerto Rico's Anolis cuvieri, and Jamaica's Anolis garmani. Cuba and Hispaniola are both home to more than one species of crown giant. 24. These are all large, canopy-dwelling species with large heads and large lamellae, scales on the undersides of the fingers and toes that are important for traction and climbing, and yet none of these species are particularly closely related and appear to have evolved these similar traits independently. 24. The same can be said of the other 5 ecomorphs across the Caribbean's 4 largest islands. In the case of the cichlids of the 3 largest African Great Lakes, each of these islands is home to its own convergent anolis adaptive radiation event. Other examples presented above are the most well-documented examples of modern adaptive radiation, but other examples are known. Populations of 3-spined sticklebacks have repeatedly diverged and evolved into distinct ecotypes. 26. On Madagascar, birds of the family Vangidae are marked by very distinct beak shapes to suit their ecological roles. 27. Madagascan mantellid frogs have radiated into forms that mirror other tropical frog faunas, with the brightly colored Mantellus mantella having evolved convergently with the neotropical poison dart frogs of Dendrobatidae, while the arboreal Bufus species are the Madagascan equivalent of tree frogs and glass frogs. The 6 xerophyte snakes of Madagascar have evolved into fossorial, arboreal, terrestrial, and semi-aquatic forms that converge with the colubroid faunas in the rest of the world. Madagascan animals are significantly older than most of the other examples presented here. Madagascar's fauna has been evolving in isolation since the island split from India some 88 million years ago, and the Mantellidae originated around 50 Maya. 28. Older examples are known. The KPG extinction event, which caused the disappearance of the dinosaurs and most other reptilian megafauna 65 million years ago, is seen as having triggered a global adaptive radiation event that created the mammal diversity that exists today. Also the Cambrian explosion, where vacant niches left by the extinction of Ediacaran biota during and Ediacaran mass extinction were filled up by the emergence of new fauna. Hawaiian lobelioids. Hawaii is also the site of a separate major floral adaptive radiation event, the Hawaiian lobelioids. Hawaiian lobelioids are significantly more speciose than the silver swords, perhaps because they have been present in Hawaii for so much longer. They descended from a single common ancestor who arrived in the archipelago up to 15 million years ago. 20. Today the Hawaiian lobeloids form a clade of over 125 species, including succulents, trees, shrubs, epiphytes, etc. 20. Many species have been lost to extinction and many of the surviving species endangered. Caribbean anoles. Anole lizards are distributed broadly in the New World, from the southeastern U.S. to South America.